Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. We are launching our GPCR consulting services. More information will be available soon. In the meantime, if you need support with your R&D project, understanding the pharmacology of your receptor, or need help getting an idea off the ground, email us at hello@drgpcr.com. We have fantastic talks coming up at the Dr. GPCR Virtual Cafe. Visit drgpcr.com/virtual-cafe to see our upcoming live events. Get your tickets today on Eventbrite. Don't forget, space is limited. To become a speaker or to sponsor an event, you can fill out the form on the main page of the Virtual Cafe. Go to drgpcr.com/virtual-cafe today. Are you subscribed to our monthly newsletter? Stay on top of all your GPCR news by subscribing today at drgpcr.com/newsletter. Also, to watch previous episodes of the Dr. GPCR Virtual Cafe or the podcast episodes, subscribe to our YouTube channel today. Last but not least, stay tuned for more information about the upcoming Dr. GPCR Summit held between September 13th and 19th, 2021. Visit drgpcr.com/summit for more information and how to submit your talk. And now, let's dive in. Hello, everyone. This is Yamina from Dr. GPCR, and I have the great and immense pleasure to have with me Dr. Michel Bouvier from the University of Montreal. So Michel is the Chief Executive Officer of the Institut of IRCM. He is a Principal Investigator in Molecular Pharmacology Research Unit, also at IRCM, and he is also a professor at the Department of Biochemistry and Molecular Medicine, Faculty of Medicine at the Universite de Montreal. Hi, Michel. Thank you so much for being here today. I'm so excited. I'm so excited to have you. So we're going to jump directly into the questions. One of my first questions is, who are you and what do you do? But I think we can skip that because everyone in the GPCR field should know who you are and what do you do. What I wanted to ask you as a first question is, how did you get into science and how did you get in the GPCR field?
Yeah, so I would say my love for science started quite early. As a kid, I was reading a lot about science, but I was reading more about, it was a time where man was planning to go on the moon. So I was very interested by this initiative of, the Apollos and all that. So that sort of led me to read about physics and chemistry and all kinds of topics. But then the real incentive came in the high school, in fact, where I had two very inspirational professors, one in chemistry and one in biology, who were really fantastic professors. And especially the chemistry professor, his name was Mr. Benovic, was passionate about chemistry and teaching. So he had us come, later at night or the weekends to run real chemistry synthesis experiments, asking us to go to university. We're high school, not even CEGEP, right? Go to university, find recipes for synthesis. And so I synthesized chlorine when I was a high school student. So that gave me sort of the idea of what it meant to do science. And then from there, it was clear that this is what I wanted to do. And I love both chemistry and biochemistry and biology. And it became clear to me that biochemistry may be a good solution because I could not decide between the two. So I did follow biochemistry training. And then after my BSc, I was very much interested in understanding more the chemical basis, if you will, of life. So I thought that going to physiology would be a good idea. And so I interviewed with a number of potential mentors. And the one who attracted my attention was Dr. Jacques de Champlain, who became my thesis advisor. And I became very interested to work with him for a couple of reasons. First of all, he was, again, very inspirational, very passionate about what he was talking about. He looked pretty, relaxed at the time. He had a beard and he had sandals, and so this was, the 80s. So, but also he was working on the role of the sympathetic nervous system in hypertension. And my father had died when I was 13 of the consequences of a form of hypertension that he could not treat.
Wow.
Very severe hypertension. And he died at 39 years old. So for me, this was sort of an incentive also to try to understand the mechanism behind such a disease. And if I could at least understand, maybe even help in that direction, it would be fantastic. So I did my thesis on this topic, which is the role of the sympathetic nervous system in hypertension. And in particular, I was interested in the role of what is called presynaptic receptors in regulating the release or the secretion of catecholamines, norepinephrine and epinephrine, and that contribute to some extent to the etiology of hypertension in some subpopulation of patients. And so, of course, presynaptic receptors happen to be G protein coupled receptors, although we did not call them that way at the time. They were, adrenergic receptors. Yeah. And so I was working on the alpha-2 adrenergic receptor at the time, and I published one of the first paper showing the role of presynaptic alpha-2 receptor in regulating the release of norepinephrine and epinephrine from the adrenal medulla, actually. So then, obviously, receptors. When the time came for postdoctoral studies, yeah. I looked, to see where I could do that. And the name that came up all the time about receptors were Bob Lefkowitz. It was Bob Lefkowitz. So I was, candid, and I just picked up the phone and called him. and later on, he told me that I was very lucky because normally he never picks up, calls like this from people he doesn't know. But he did. So he talked to me, and then later on, he also told me, you look so enthusiastic and so, voluntary to do some work that he invited me to come down, and we had an interview. And after, let's say 30 seconds in the interview, I knew this is where I wanted to go because, again, I found this passion, I found this enthusiasm about science, about knowledge. And Bob is a very charismatic character. Yeah. And so I knew that I wanted to go there. So I did my postdoc in Bob's lab, worked on phosphorylation of receptors under the guidance of, in particular, Jeff Benovic, who's also now, one of the major names in the GPCR field. And this was excellent training for me. And I had the chance to be there at the same time as Brad Kobilka and Susanna Kotekia and a lot of the names that we all read now. Yeah. So it was a great period. It was a fantastic team at the time. So I learned a lot while being there. And I had the chance to do among the first, maybe the first experiment of heterologous expression of a GPCR, it was not HEC293 cells at the time. They were, CHOs and CHW cells. But looking at the role of different parts of the receptors, doing site-directed mutagenesis and identifying the phosphorylation sites involved in desensitization. So I became very, very much interested in understanding the molecular mechanism regulating signaling efficacy. And then after finishing my postdoc, I had a number of offers from different places in the US, in Canada. And with my spouse, we decided to come back to Canada. And I joined the Universite de Montreal, the Department of Biochemistry, and started my own lab on GPCRs, in particular on the mechanism controlling signaling efficacy.
So just a side note, I love the fact that you picked up the phone and called Bob Lefkowitz. I had an interview with Arun Shukla, who was at the time doing his PhD in Germany, and he wrote an email to Bob, and Bob said, just come on over, let's chat. So I'm trying to gather all these people who were in Bob's lab and see how they got into Bob's lab. But before we move forward, I want to take a step back. So you got interested in science in high school. And then what happened between high school and, your bachelor's degree at the University of Montreal? It is of note that you went to the Bois de Boulogne College, where I went as well. And I also did my bachelor degree in the Department of Biochemistry. What kept you going into the direction of science?
Well, at the CEGEP, this was an interesting CEGEP because it used to be a college run by, religious. And so it was a small CEGEP, and especially the pavilion where the science people were, where I was very small, very cozy. And so it was a fun time. CEGEP is a fun time. So, yes, I like the chemistry courses. I like the biology courses, but quite frankly, my memories of CEGEPs are not very much related to academics. They're more related with, the music room and the chess room, and we were playing chess and we were, playing music, and I was involved in the journal of the CEGEP. So it was more like, becoming an adult, right, than anything else, I think, CEGEP. So But I loved it. It was a great place. But I cannot say that this was influential in going one way or the other in terms of my career or my decision. Then biochemistry, when I registered there, as I said, I thought this would be the good place for me, and it turned out to be the good place for what I wanted to do. The training was great because there was lots of, as I said, I like both biology and chemistry, and there was quite a bit of chemistry in the first years. Not that much biochemistry, actually. Biochemistry was coming only later during the training, but for me that was okay because, as I said, I was very much interested in the chemistry aspect. So my time there was great. I did relatively well during these 3 years in terms of academics records and all that. And the labs, I love the labs. I love working in the lab. I love the idea that we were working in teams in the practical work. And so it was just like, for me, the goal was always to go to grad school. From the day one, there was never a question that I would not go to grad school and become a scientist. I guess these were easier time also. Science was thriving. This was the expansion phase, and the funding was adequate. So for me, there was no doubt. So it was just a process that needed to be done to finish my bachelor's degree. I enjoyed it. There was no issues. Mm-hmm. But again, this was not where the orientation came. The orientation came when I started looking for a mentor for grad studies, where both personal, as I mentioned, both personal history and the charisma and the enthusiasm. And you'll notice that I speak a lot about enthusiasm and passion, because I think that to be successful in science, this is one of the most important ingredients: to love what you do, invest yourself entirely in what you do, and because there are difficult times in research, as everybody knows, and if you don't have this passion and resilience, sometimes it's going to be too difficult maybe for someone.
It can be. And I think it's a matter of getting through the tough times, because every time you get to the result or to the answer that you were looking for, you get that dopamine shot, and that carries you from cloud to cloud, or from great data to great data.
One thing that I used to say to the student, and so there's a couple of things. First of all, when you do something in experiments and you get the result, and this experiment has never been done before, well, you know what? You're the first one seeing that. You're the first one seeing the results. So you're the first one understanding a little bit more how things work. And life is a lot about that. Forget science. Life is a lot about understanding how it works, and growing into understanding this better and functioning in such an environment. So there's similarities between growing into life and growing as a scientist. So this is one thing. The second thing is this idea of traveling, of a trip. For me, life or a scientific endeavor is a trip. Of course, the goal is important, but for life, the goal, is death. So the end is death. So not that exciting, depending on your beliefs, but the traveling is exciting. The traveling is great. So doing research is, yes, of course, getting data and getting the results. That's very important, and the ultimate data. But all the small pieces of data that you get along the line, and understanding, is that exactly what we thought? Is that different? If it's different, how can we change the hypothesis, modify the way we're thinking about it? And also, just the last thing on this is something that Jacques de Champlain was telling me, my PhD mentor. This is a citation from Julie Axelrod, that Julie Axelrod was the mentor of Jacques de Champlain, a luminary, was working at the NIH, as well, Nobel Prize winner. Yep. And Julie was saying that when you have a data that you're expecting, it's okay. You were expecting it and you got it. It's nice. But when you get a data that you did not expect, and you rule out the possibility this is an artifact, well, now you have something. Now you have a real discovery. But you have to be, smart enough to understand what it means and act accordingly. And one sentence I often say to my students is that, oh, this is great. I like this data. We're not smart enough yet to understand exactly what it means, but let's work on it. Maybe we can. So that's the idea.
I love it. I love that. And I think you're right in the sense that when you get something that you were expecting, well, good. it's important to get that. But when you get something that you didn't expect and it raises more questions that it answers, that's when you get into on a track that can, really you can make great discoveries out of there. So you're in Bob's lab, you work on adrenergic receptors. You're one of the first, or with the first person who actually expresses a receptor in CHO cells, not in HEX cells. I think this type of experiment is now such a routine thing that master's students can do it. But how did you feel when you finally were able to do this type of experiments in Bob's lab?
Well, it was incredible because the reason I went to Bob's lab is because after my PhD in physiology, I liked it. I liked it a lot, actually. But, it was observational. It was like, almost phenomenology. We do this happens. But the mechanics behind it was not clear. And that's what I wanted to understand, was the mechanics behind how these things really work. And I arrived at Bob's lab at the perfect time, because it was the time where the transition between pure biochemistry, where they were purifying the receptors, doing reconstitution assays, identifying biochemically the sites, and the molecular biology era. So it's just that junction. And so the cloning of the receptors was happening, and all these tools that now allowed us to really look into the fine details of what these receptors were and how they function. This was exactly that period. So I felt great because I was part of this, even if I think I did some contributions, but even without these contributions, just being there and witness that was already rewarding. Just appreciating that we were changing era, that we're getting these fantastic tools. And I remember having a discussion with Monsieur Bouchard, the previous Prime Minister of Quebec, in a social event. And I was telling him that, and this was not that very long after I came back from training from Duke. And I was telling him that for life sciences, we're living times that were very similar to the time of the moon conquest. So I'm back to this because the tools were finally there. We could really get into the nitty-gritty of how they function. So that's how I felt, and I could see it at the time. It's not just retrospective thinking. It's I could see that this was happening while we were doing it. So this is how I felt.
That's amazing. So you've worked on adrenergic receptors. Do you have a favorite GPCR? Because I know that you've worked on many different receptors. There's arm-long set of publications, but is there a favorite, or is there a family of favorites?
No, in fact, it's like kids. You cannot have your favorite kid. So I don't, quite frankly, and I've never looked at research that way. I know some people spend their entire life working on one receptor, and then that's what they do. I've never looked at it that way. I always looked at it by, what is the question I would like to tackle? Mm-hmm. And again, this is a lesson I learned from Bob. I think he had learned from somebody else, which is always the case in science, right? Which is like, give me the right model and I'll find the answer. So for me, it's the question that drives the program. What are the questions we are asking right now? And so depending on the questions we're asking, let's find which receptor is the most adequate to answer it, or let's try to find out which of the receptor has a role in this phenomenon that we want to study. So I've never had this, some people think my favorite is the beta-2 adrenergic receptors because that's the one I published on the most, and this is where I started, both in my PhD, I was working on adrenergic receptors, both the alpha-2 and the beta-2 actually. And then in Bob, it was only beta-2 when I was there in Bob's lab. And when I came back, I focused on this one again because I knew it more and we had the tools. But as we evolve, with the more tools and all that, I've been working, as I've been working on the chemokine receptors, on the vasopressin receptor, on many of them, even on class C receptors. I have a couple of papers on the GABA receptor, and in particular because of the dimerization story, I became involved into these receptors as well. So it's always a matter of questions and tools, really, for me as choosing which receptors we're going to be studying.
I like that. So now the entire GPCR field who listens to the podcast will know that the adrenergic receptors are not necessarily your favorites. Now we'll know. So you're talking just a minute ago about tools, and we cannot talk to Michel Bouvier without talking about BRET. Mm-hmm. Can you tell us a little bit more on how you got to a point where you learned about BRET, or you started developing BRET tools to better understand GPCR function?
Yeah. So the BRET story is an interesting story because it involves several people. So this came from observation that Terry Baer did when he was a postdoc in my lab. Among the first evidence suggesting that GPCR may form dimers and oligomers based on biochemical aspect. And so I'm going to mix two stories at the same time. Right? I'm going to mix a bit the story of the dimer and of the BRET. So this was, and still today for the family A is still somewhat controversial. At least the role. I think the fact that they can associate is less and less controversial, but the role of family A dimerization is still a matter of interesting question actually, which we are still working on. So Terry had these data with covalent precipitation and interfering with peptides, and we tried to publish that in the high impact journal at the time. And the journals told us, yes, it's nice, but you need to show that this exists really in living cells. So we decided that yes, we needed to do that, but it would not take the 90 days or 60 days of revision to do that, right? You would need to develop a tool. So we submitted the paper somewhere else. We submitted it to GBC actually. And then we started thinking about what could we do to study that in living cells. And I was aware of FRET, fluorescence resonant image transfer, but I had read about the fact that there was marine animals that had this natural phenomenon of transfer of energy between luciferase and GFP. And also people at a small biotech company in Montreal, which was called Biosignal at the time. And Michael Dennis was there. And he had also started reflecting on this. And Michael was a close colleague, but a friend. And so we started discussing this, that maybe we could use this technology which occurs in in marine animals naturally, to do a proximity-based assay that could allow to address questions like this. And he had at the time someone in the lab starting thinking about that, whose name was Eric Joly. And there was an employee back then there, which was Stéphane Anger. And Stéphane wanted to do a PhD. He didn't want to stay as a technician. And I interviewed him because he wanted to come to the lab. And after a couple of interviews, he was a hockey player and very determined and very, go get it. So I like this aspect of his character. So he came to the lab and he started working on this concept of BRET. And the rest is history, because then it worked nicely. We could develop these tools, and at the same time, so good ideas, very many people have them at the same time. At the same time, there was the group of Piston in Vanderbilt, who also had the same idea, but in bacteria. They were working on clock proteins. And so the two papers came out, I think, a month or two apart on the first description of BRET. And there's also a lesson there in the way I publish and the way I should maybe not publishing. So this first paper, which was published in PNAS on BRET, we had, in this paper, we had BRET used to look at dimerization of a GPCR. We had BRET used to look at the activity of caspase. And we had BRET used to look at recruitment of beta-arrestin. All of that in one single paper. And of course, this paper is cited for BRET. It's cited for dimerization, but never, ever cited for the caspase assay or the beta-arrestin assay. People are citing other of our work on the beta-arrestin assay, but that came much later. So that tells you, sometimes the way that you present things makes, decide how these papers or these discoveries will be seen by other people. But this is the story behind this. And then we saw the potential of BRET in terms of generating what we call biosensors to monitor different type of activities in cells, and in particular the first one we wanted to look at was the G protein themselves. And Céline Galès was in the lab at that time, and we had made some attempt before she joined with no real success. And when she came in, I told her, and very often when something doesn't work in the lab, it's very difficult to convince somebody else to jump. And so until then, it didn't work. And so I told her, I said, Céline, I think I have the perfect project for you. This is a great idea, but it looks like nobody right now in the lab is able to get it to work with the G protein. So would you take this challenge? And Céline, being Céline, she said, yeah, I'll do that. And within a matter of months, she got it to work with the G protein. So again, the importance of the passion and the determination in science. And so the G protein was the first one, but then we developed it more and more and more. We created consortium around it with other researchers in Quebec, including Thierry Hébert, Stéphane Laporte, who made very, very important contribution to the more recent generations of the BRET sensors with us. Also Richard Leduc in Sherbrooke, Graciela Pinheiro at Saint Justin, and we work still very closely together to develop more and more of these biosensors and use them to answer different questions.
I love it. Yeah, and I know Céline, and I've worked very closely with her for a short period of time when we were testing her G protein BRET construct with chemokine receptors. And you're right. When Céline says, "Sure, I'll do my best," she'll get it done. She'll definitely get it done. And it's fascinating how from a conversation, an idea and a question that came back from a reviewer on an immunoprecipitation paper, we are where we are today with so many biosensors.
Yeah, but I'll tell a story on this about publications. I have my issues with the way that publication system is working nowadays. But so when we got it right, we got it to work.
Yep.
The BRET, and so we sent this new story with dimerization of the beta 2 with BRET to the same high impact journal, which I think was Nature. And they came back and said, well, no, now it's old story, because there was this paper from Lakshmi Devi, good paper on opioid receptors, but there was co-precipitation only. There was no in vivo stuff or cell-based, living cell. But now it was too late, right? The thunder was gone. So it was still published in PNAS, which is good. But just tell you that, the way that the science evolves is an interesting sociological topic for. It's another aspect of life which is interesting, sociology and the sociology of science. I think it's fascinating.
Yeah, that's phenomenal. So the idea is that you don't have a specific GPCR that you are infatuated with. You're looking at the question first, and that's when you try and put in the specific tools, including the receptor, that will allow you to get an answer. How do you see the relationship between understanding GPCR function and drug discovery? Now, with all the tools that you have and you're trying to answer these questions, where do you think those fit in the drug discovery department, let's say?
Yeah. Okay. Well, that's a very important aspect. This is always when it was in the background in my lab, which is let's try to uncover basic mechanisms. By the same token, develop tools that can allow us to answer the question the best, and use these tools, if possible, to drive drug discovery programs. And there's a couple of examples which happen in the lab, and I'll come back to the more recent one after. But the first one, really, in that realm, was a collaboration with Stefano Marullo. Again, coming from a meeting, both of us. Stefano is a very good colleague and a great friend. So we were in a meeting together, and we hear this talk of people using the HIV body to produce protein in insect cells. This was the idea, and so we had this idea that expressing GPCRs in insect cells could be a very good thing to look for. Biochemistry, high quantity. This was before crystallization had occurred.
Yeah. Also
pharmacology, membrane preparation. And again, with Michael Dennis, which was not in the company at the time, we had decided that maybe we should try to produce GPCR in baculovirus. And so we went and were successful in producing the GPCRs in baculovirus. And in fact, we this was patented and it was licensed by some companies to use. And then it led to the creation of Biosignal, the company that I was telling you before, who their first, product was selling membranes from insect cells expressing different GPCRs. So from very early on in my career, this was always the idea. So when BRET came, the other discovery that we made in the lab, probably one of the first lab, if not the first lab, was this idea of functional selectivity or bias signaling, where ligands could activate the different pathways. And this came on the heel of our discovery of inverse agonism, where some compounds could inhibit spontaneous activity. Again, a discovery that was made exactly the same time as the same observation in Bob Lefkowitz's lab. I wasn't there anymore, so we were colleagues, but a bit competitors. So this notion now that receptors don't have only two conformation, and they can be inhibited, they can be activated, and they can have different activity towards different effectors, became for me quite an interesting possibility for drug discovery, because indeed maybe only some of these pathways that can be activated by a given receptor would be of therapeutic importance, and maybe some of the other ones could lead to adverse effects. Now I think this concept is fairly well accepted. This was not the case back then. And even the inverse agonism, when I first, I have a memory of this. I was presenting this in a drug company in France. And I was presenting the concept of inverse agonism, right? A compound that can inhibit the spontaneous activity of a receptor. And half of the crowd were chemists, and half of the crowd were biologists, pharmacologists to be more precise. And so while the pharmacologists were ready to burn me for heresy, the chemists were saying, "What's new, pussycat? That's allosteric regulation." And so it was very striking, again, in sociology of science, how these two things, but the pharmacologists would not have it, right? Because they had this notion in their head of the key and the lock, and that the lock is locked, and then when you have an agonist, you turn it on and it goes, and the antagonist is the chewing gum that you put in the lock. And these images are useful to understand things, but then they should not get into the way of understanding how it really works, if they are just models. So the same thing happened when we went to functional selectivity, and people thought that this was just artifacts of the acid that were used and all that. And that's why we wanted to have homogeneous assays. So testing all of them using the same type of assays, not with different level of amplification, not with different type of reagents, if you will, to do it. And that's why we went to develop more and more the BRET assays to look at the potential for drug discovery. And it became obvious after a while that it was important. And now we have examples, although the proof of a new drug that is much better because it's biased still needs to be born. But I think there's clear evidence in preclinical data that one, bias does exist, or functional selectivity does exist. Two, that it may have important physiological outcomes in terms of drug action, at least in animals. And now the next level, and I think there start to be some example by Trevina and others that for even for treatment, this could be an important aspect. And so we are collaborating with different small companies and bigger companies to help them in their drug discovery program using these tools and progressing towards new functionally selective compounds that could have clinical importance.
Thank you for that. I want to take two little steps back. So you were talking about having these meetings to try and figure out how we can use BRET as a technology to better understand, protein-protein interaction or, GPCR function. And it may be a naive question, but the way I see it is, in a dark room, in a leather sofa with a drink, you people talking about this. How do these meetings happen? Just for the audience to get an idea on how do these important conversations that ultimately lead to so great tools and so magnificent discoveries happen.
Yeah. There's all kinds of sources of these types of eureka moments or strategic decisions that are being made in different programs. And I remember one occasion. This was a meeting with Stéphane Angier and Nelly Salapour, and this was the early days of BRET in my lab. And Stéphane and Ali were a perfect team, and they're still very good friends, and both now are at the University of Toronto. And we had this discussion in my lab, and I was telling them, that these are types of data that bring tears to my eyes. And then we're trying to understand, how we could solve, specific question. And it became extremely, how can I say that, almost emotional in terms of, how we were appreciating the data and moving forward, and came up with new experiments that we would do. And this was with a very small group, right? Just Ali, myself, and Stéphane. That's all. So the three of us. Some other times it will be, as I mentioned, with the anecdote about the baculovirus and how to express the proteins into SF9 cells and have them secreted also, because we could express them in the membrane of the SF9 cells. But with Stefano, the idea we had is that we could have them secreted into the viral particles generated. And that's what we did with Stefano. But in that case, it was in a meeting, right? Stefano and I listening to a talk, discussing this talk, coming up with this idea that, let's do it, going back to each of our labs, Stefano in France, me in Canada, and getting it done. Sometime with Michael Dennis on the first part of baculovirus, which was expressing them in the membrane. He was coming back from a meeting with Michael. I was giving him a ride, driving back from the meeting to Montreal. And Michael said, what we've done on the baculovirus, we'd like to create a company out of that. And I said, well, not me, but you should create a company. I will help you. And so, so that's another, and that's why Biosenol was created. So, and some other time, it will be in a lab meeting, right? A good example of that was the discovery of pharmacological chaperones. So, as you know, we were lucky enough to discover the concept of pharmacological chaperone many years ago, working on mutations on the V2 vasopressin receptor that lead to nephrogenic diabetes insipidus, a very rare disease where patients cannot concentrate the urine. And so when I was collaborating with Daniel Bichet, a nephrologist at Sacré-Cœur Hospital, Daniel had called me and said, Michel, we have these patients which have mutations in the vasopressin receptor, and it leads to this disease. I heard that you know something about GPCR. Again, this was quite early in my career. And maybe you can help us. I said, yeah, sure, you can help us. And I thought, this is great. Nature has worked for us, right? Nature has done mutations, and we'll find all kinds. And this was way before structural biology time. So this will allow us to identify which region the receptors are important for binding, which regions are important for coupling to the G protein, because they're all loss of function mutation, and therefore we will understand how it works. And Jean-Pierre Morello was a PhD student in the lab at the time, started testing the effect of this mutation on this. And first he was very happy. He said, they all lose binding. And I asked him, okay, but do they go to the plasma membrane to begin with? He said, I don't know. I said, well, go back and check it out. So he checked it out. And of course, none of these mutations were making it to the cell surface. So now we're at the lab meeting. And this poor Jean-Pierre, his PhD student, right? He has no project. It's all retained inside the cell. These mutations are just killing the receptors, right? So what could we do? And then in the lab meeting, and I think it was Stéphane Angier, but one of the students or another person came up with this idea. We use compounds, antagonists, to stabilize receptors when we purify them, right? Maybe if we were to have a lipophilic antagonist that could penetrate all the way to the endoplasmic reticulum, maybe it could stabilize a near-native conformation. Most people in the lab hated this idea. They thought this was crazy. I loved it. I thought this was great. This was a great idea. And so, I don't know exactly how this idea came up, because that's the beauty of it. It's a community thing. Right? Someone says something and another one says something else, and then it came up. So I called my friends at Sanofi in France, and I told them, do you have a V2 antagonist, in your drawers? And said, yeah, we have that. And they said, this lipophilic. Yeah, that's one of our problems, too lipophilic. Okay. So they sent it to us, and we tested it. And Jean-Pierre tested it, of course. And here was the discovery of a compound that could rescue the expression of this misfolding mutation. So, and this one came out of a discussion at a lab meeting with 25 people around the table, maybe with one person first giving the first, spark, and then the fire caught in and moving forward. Yeah. So different ways of these ideas. Yeah.
That's really fantastic that, these compounds were in a drawer somewhere at Sanofi, and they said, we don't need it because it's too lipophilic. and it ended up
Well,
being.
it was not that bad because some of them made it to the clinic, but they were lipophilic.
It wasn't optimal for them, and it ended up being a great tool. All right, so we're almost out of time, and I know that you have a hard stop at 10. Just to recap a little bit, we started out talking about the passion for science and the passion for finding the right question and using the right tools to answer those questions. What would be your advice for junior scientists who want to contribute in the field?
Well, I think the first thing is find what you love. I think that's the first thing is to develop your own scientific personality. What kind of questions are really driving you? What kind of questions makes you wonder, how could I solve that all the way until you get to bed, And so this is important because you're going to spend a lot of time and energy on this question or these questions that you're going to be developing. So it needs to be driving you. So I think that's the first thing. The second thing I think is don't take no for an answer. I mean, there's always a way around a block. So just go after what you really want to do. And take all the means that needed, any legal means needed to do it.
Obviously.
And working hard. I mean, there's no other solution. There's a few people. Another anecdote with Julie Axelrod and Jacques de Champlain. So I always been working quite hard, and Jacques my mentor thought I was working too hard. And he was saying, Michel, you have to take some time. Working too hard. He says, look, Julie Axelrod was coming at work, with the newspaper under his arm, staying at work, 8-9 hours, leaving with the newspaper under his arm, no work at home, and look, he did great discoveries and he got the Nobel Prize. And I answered to Jacques and said, yes, but he was extremely, extremely smart. I'm hardworking. It's a caricature, but it's very rare that some people be so smart and so different from others that and especially nowadays where it's driven by technologies and understanding the different techniques and being able to master them. I think you have to be prepared to work very hard, and you have to enjoy the rewards of discovery. That also is very important. I mean, when these rewards come, let's take the time to rejoice and enjoy them and talk about them and reflect on what it means, not only for the project or the paper or the thesis, but what it means in the more global picture, the more global history. One thing I also say often to the student is that we are privileged. We live in a time where societies are paying people to understand how it works. Yeah. This is a rare time in history. People were paying of their own money to do that back in the Middle Age, unless they had a king that could, help them. But this is rare. And so even if we don't make a big contribution, just being equipped to understand the big discoveries is amazing. This is a great reward. And you need to appreciate that so that you can put the hard work that you need to put in.
Great. Thank you so much. So you were mentioning that, Professor Axelrod was really unique and a really smart person, and you're hardworking. But let me tell you, I've seen you sit 8 hours at a conference and always be on point and ask the right question. And I always found it fascinating how you were able to focus through all those talks from various topics. So if Professor Axelrod is at the top where the way you look at it, you're definitely close to that for me. I never got to tell you this, but I always found it very fascinating. And even if it was a student presentation, we had, the competition, the whole day competitions, the way you asked the question after, the talk, even if the talk wasn't necessarily, it was a student talk, I always told myself, how does he do it? How does he focus for such a long time and also ask the right question?
Well, because I love it.
Well, then there you go. Now we have our answer. So today what we learned is that you don't have a favorite GPCR. you work hard, you want to make discoveries, and ideally get those discoveries into the clinic and help patients. And you are as motivated as you were in high school and as you are the same today. So thank you so much, Michel, for your time. I'm very, very excited that we got a chance to chat, and I'll let you go. Thank you so much again.
Merci beaucoup.
Merci.
Thank you.
Au revoir.
Bye-bye.
Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, and Jin Chung. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. Another way to support us is to share the podcast, the newsletter, and the virtual cafe with your colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.